continuing with diseases of the immune system, and then we will discuss if this immune system do not work properly, it can result in autoimmune diseases. Autoimmune diseases. Now, what is defined as immune reaction against self-antigen, and it results from breakdown in the self-tolerance or failure of self-tolerance. What is self-tolerance? Self-tolerance is a normal state of non-responsiveness to one's own antigen. So, autoimmune diseases results from failure of self-recognition and failure of self-tolerance. Now, tolerance is further is said divided into central tolerance, in which death of the self-reactive lymphocyte take place in thymus during positive and negative selection, and when these T lymphocytes from thymus and B lymphocytes from bone marrow enter into the peripheral circulation, the peripheral tolerance take place through energy, that is the masking or the loss of reactivity of T and B lymphocytes to a own antigen, and suppression by regulatory T cells, deletion by apoptosis, sequestration or antigen masking, that is like they mask their antigen, and this is also adapted by certain like tumors and different kind of pathogens. They also adapt this phenomena to escape from this being killed by the immune system. Strong genetic predisposition also leads to through the MSC, and often related to other autoimmune diseases. This autoimmune disease is often triggered by certain infections of pathogens like post-streptococcus glomerulonephritis. So they also they have their role in certain autoimmune diseases. Tolerance. Tolerance is defined as immunological non-reactivity of a body to an antigen, or our immune system to an antigen. It results from a previous exposure. The most important form is non-reactivity to self-antigen. When an antigen induces tolerance, it is tolerogen. Now this diagram shows the reaction when our normal immune system, it comes like B cell with attached immunoglobulins on its surface, or come in contact with immunogenic antigen. It either immunogenic antigen, the B cells will proliferate and release their machinery immunoglobulins into the secretions, and that differentiation through plasma cells, and that will result in killing of that immunogenic antigen. Now, when intolerance, what happens when this B cell with attached immunoglobulins on its surface come in contact with tolerogenic antigen, like either it's result in energy, functional inactivation, that there is not activated, for example, activation of the T of cells required co-stimulatory molecules. That co-stimulation do not take place, and the cell become functionally irreversible function in unresponsiveness or inactivation take place, or either the cell will be die or death take place through deletion and apoptosis. So there will be no response. Tolerance mechanisms is through B cell tolerance in bone marrow, and when the B cells enter into the peripheral circulation, it takes place through deletion, energy, antigen and receptor editing. T cell tolerance in thymus take place through deletion and ignorance. loss of T cell regulatory T cells, anti-idiotype antibody. Now, thymocytes, when they arises from bone marrow, these immature thymocytes enter into the thymus, and they have their rearrangement of the TCR take place, like T cell receptors, and they have both CD3 and CD4 receptors on their cells. Their positive and negative selection take place in the thymus. In positive selection of the cells where receptors TCR binds with MSC molecules, and when these bind and recognized by MSC, they results in and they do not interact with self MSC. They pass the test and they enters into the peripheral circulation as CD4 helper T cells or CD8 cytotoxic T cells. Negative selection also takes place at that time, and death of the high affinity receptors with self MSC results in activation of macrophages and dendritic cells and killing of the self-reactive T cell take place. Now, death at that time also take place through apoptosis if the cells do not interact with MSC molecules. If the TCR receptors fails to interact with MSC, they also result in apoptosis and of those thymocytes of T cells. So tolerance or T cell tolerance that take place in thymus is through positive selection and negative selection. Positive selection is that the cells that are capable to recognize and bind to self MSC through peptides like plus M and MSC molecules, co-stimulation also take place, are selected to grow. Negative selection is that cells that recognize and bind with self peptides are autoreactive cells, and they undergo apoptotic cell death because they are harmful to the body. Cells that pass both positive and negative selection are graduated from thymus, and they enter the circulation as mature T lymphocytes, as helper T, CD4 T lymphocytes, and cytotoxic CD8. Mechanism of tolerance: clonal deletion that results in thymus and bone marrow. Autoreactive T cells in thymus are eliminated when they interact with self-antigen during their differentiation. This is called negative selection. And a protein called autoimmune regulator, it plays their role in inducing the self-antigen expression. And if some mutation take place, this in protein AIRE, autoimmune regulator protein, it results in certain autoimmune diseases. Likewise, the differentiating early B cells, when they become tolerant when they encounter self-associated antigens in the peripheral circulation. If they react with their self-antigen, some changes will take place in their immunoglobulin machinery, and this the reactivate the machinery or rearrangement will take place. Or if that do not take place, that B cells will die by apoptosis. So if this clonal deletion of self-reactive B and T lymphocytes do not take place properly, that will result in autoimmune diseases. Second mechanism of tolerance is through clonal energy. Energy is defined as functional inactivation, that is irreversible, and it occurs when T cell recognize or B cell recognize self-antigen. Autoreactive T cell, when they are exposed to antigenic peptides, co-stimulation requires two signals. So they lose their second signal and they become allergic to that antigen. B cell, when exposed to large amounts of soluble antigen, they downregulate their surface immunoglobulin IgM, and they become anergic or functional. They get functionally inactivated. This is antigen-presenting cell or a macrophage that have MSC attached on its surface, MSC2, and when this MSC interact with the T cell, that is with the TCR, T cell receptor, this is signal 1. And other for activation of T cell, second signal is also required, that is when it interact with like B7 receptor with CD28. So if this co-stimulation do not take place, T cell will not be activated and it will not result in like immunologic reactions or killing of that antigen do not take place. And if it fails to receive the second signal, it dies by apoptosis. Ignorance. It can be shown that there are T and B cell specific for autoantigens that are present in the circulation. These cells are quite capable, T and B cell, they make a response but are unaware of the presence of these autoantigens in the circulation. How this arises? There are two reasons. First is that the antigen may be in circulation, that is in too low concentration. Since all lymphocytes, they have a threshold for receptor components. If that is not required to trigger a response, but if there is concentration of antigen is too low, receptor that will not be sensed by the TCR receptor. Ignorance or antigen sequestration. Second possibility is more interesting one, that some antigen are sequestered from immune system that are present in locations which are not freely exposed to surveillance. These are termed immunologically privileged sites such as eye, CNS and testes. So any physical trauma that results in sequestration of these antigens from those sites can lead to autoimmunity. Fourth mechanism is through receptor editing. B cells that encounter large amounts of soluble antigens in the body and bind to this antigen with the very low affinity, they become activated to re-express their genes like RAG1 and RAG2, and there will be rearrangement of the immunoglobulin machinery within the B lymphocytes, and they become tolerant and will to those antigens. These genes can, like RAG1 and RAG2, cause them to undergo DNA recombination and change their antigen specificity. Mechanism of tolerance could be through anti-AUTOIMMUNE antibodies formation, which are produced in the process of tolerization. They prevent the receptor from combining with antigens, so inhibit immune response to that antigen. And suppressor T cells. Now during clonal deletion, if the cells they do not die by apoptosis, they differentiate into suppressor T cells or regulatory T cells. These T cells when come to low and high doses of antigen, these suppressor T cells then suppress the immune response of both B and T lymphocytes. They express CD25 receptor and FOXP3 transcription factor, and that inhibit the lymphocyte activation, and effector mechanisms are also activated through release of certain cytokines. If the mutation in FOXP3 receptor take place, it can also result in certain autoimmunities. Autoimmunity is defined as immune recognition and injury of cell tissues. It results from failure of self-tolerance or loss of salt tolerance. Autoimmunity is it's the breakdown of mechanisms which are responsible for self-tolerance, that is central tolerance, clonal selection and peripheral tolerance. or induction of an immune response that against components of the cell take place, that is through certain microbes that induce certain immune response and cross-react with body on tissues or bone, that results in disruption and immunologic reaction against the body tissues. Loss of self-tolerance. Most self-peptides are presented at levels that are too low to engage affected T cells, and when they are presented at higher levels, they induce clonal deletion or energy functional inactivation. Autoimmunity arises most frequently to tissue-specific antigens, which have certain MSC molecules attached on their surface, and they present the peptide at an intermediate level that are recognized by T cell without inducing tolerance. So certain autoimmune diseases have association with MSC. It depends upon the level of presented autoantigenic peptide, and if that is determined, and it's also determined by residues in MHC molecules, a major histocompatibility complex that are expressed by human leukocyte antigens. These molecules of glycoproteins, they govern the affinity of peptide binding. So, autoimmune diseases are associated with particular MSC genotype. This shows certain classification of the autoimmune diseases and their target organs and the tests that can be done to identify those autoimmune diseases. Certain diseases are organ specific and others are systemic, that they involve all body system and they're not organ specific. Organ specific like Hashimoto's thyroiditis, which involve thyroid gland, and antibodies are directed against thyroglobulin, and the diagnostic tests are like it can be heme agglutination or serum assays for antibodies in the body. Primary myxedema is also involves the thyroid, in which the antibodies are directed against the cytoplasmic TSH receptor, and immunofluorescence can be, through immunofluorescence, these antibodies can be detected. Pernicious anemia, in which the antibodies are directed against the red blood cells and or intrinsic factor that are that organ specific pernicious anemias affect the RBCs, and they are directed against the intrinsic factor or gastric parietal cells, and they interfere with the absorption of vitamin B12. A diagnostic test is B12 binding to immunoIA fluorescence. Addison's disease, it involves the adrenal gland and adrenal cells. Antibodies are directed against those cells and can be detected by immunofluorescence. Male infertility, the main organ, that is sperm, and antibodies are directed against spermatozoa and can be detected by immunofluorescence. Insulin dependent juvenile diabetes, in the organ that is involved as pancreas, and antibodies are directed against pancreatic beta cells. Insulin resistant diabetes is a systemic disease in which antibodies are directed against insulin receptors. Myasthenia gravis, it basically involves the muscles, and antibodies are directed against acetylcholine receptors or neuromuscular junctions, and immunofluorescence can be done for that. Vitiligo is basically involves the skin and joints, as well as in which the antibodies are directed against melanocytes and can be detected by immunofluorescence. Rheumatoid arthritis involves systemic disease, skin, joints, kidney, in which IgG antibodies are directed different organs of the body, and IgG lattice agglutinin can be done to confirm the disease. SLE is also a systemic disease, systemic lupus erythematosus, and it involves the joints more commonly, and skin and other body organs, kidneys, as well as, in which the antibodies are directed basically against the nuclear proteins, DNA and RNA. And immunofluorescence specific antibodies like anti-RNA DNA can be detected through lattice agglutination. Classic autoimmune disease, as have discussed, like they are systemic. They mostly involve not a single tissue or organ, but it involves different body parts or different organs, like lupus, SLE, systemic lupus erythematosus, rheumatoid arthritis, and Sjogren's syndrome. It's not also specific, not only involves the salivary glands. It's a systemic disease. It involves other tissues as well, as systemic sclerosis, or that results in connective tissue and fibrosis all over the body, skin, esophagus, and other body organs, as well as mixed connective tissue diseases and polyarthritis nodosa, periarthritis. Classic autoimmune diseases which are local, like we have discussed, Hashimoto's thyroiditis, autoimmune hemolytic anemia, in which antibodies are directed against blood cells, multiple sclerosis, and antibodies are directed against myelin, autoimmune orchitis, and Goodpasture's syndrome, in which autoantibodies are directed against in kidney, the glomerular basement membrane, and in lung, autoimmune thrombocytopenia, in which autoantibodies are directed against platelets, pernicious anemia, in which the antibodies are against intrinsic factor that interfere with absorption of vitamin B12 and that results in anemia. Insulin-dependent diabetes mellitus, against beta cells of islets or beta cells of pancreas, myasthenia gravis, and Graves' disease we have discussed in the. So autoimmunity, how it develops, mechanisms are basically through antibodies and effector or regulatory. Autoimmunity etiology, sequestered antigen. Sequestered antigen lymphoid cells, they then may not be exposed to some self-antigen during their differentiation that are present at certain sites like testis, brain, and eye, and their body get exposed to those antigens when physical trauma takes place, or they enters into the peripheral circulation that results in orchitis and uveitis. A release of antigen from those organs result from surgery or accidental traumatic injury. And it results in stimulation of an immune response, and that is initiation of an autoimmune disease. Second cause, it could be loss of suppressor T cells, escape of autoreactive clones. The negative selection in the thymus may not be fully functional to eliminate all the self-reactive cells. Not all self-antigens may be presented in the thymus. Certain antigens may not be properly processed and presented in the thymus, so they escape through the check system and that enters into circulation. Cross-reactive antigens. Antigens on certain pathogens may have determinants which cross-react with self-antigens, and an immune response against those determinants may lead to effector or T cell response or antibody response against tissue antigen, such as post-streptococcal glomerulonephritis and carditis, anticardiolipin antibodies during syphilis. And they have association also like with Klebsiella and ankylosing spondylitis. Diagnosis is clinical, but detection of antibodies against reactive or soluble antigens by ELISA can be done. Detection of antibodies against specific tissues and cells by immunofluorescence can be done to detect autoimmune diseases. But in certain cases like biochemical assays in Graves' disease and pernicious anemia, serum assays can be done. Treatment is basically anti-inflammatory or drugs like corticosteroids, immunosuppressive therapy, or certain anti-etiotype antibodies can be given to the patient.